Streaming. Hello, hello, hello. I think I'm on the wrong channel. So if I go over here, this is wrong. RyanReardon71. Allison, can you hear me? You're watching that, buddy?
Yeah.
So you can see me behind there.
Yeah, but you're like delayed by a few seconds.
Okay. Maybe I need to update this screen then.
You're delayed by roughly 7 seconds.
Okay. How do we update this page?
And you're on your regular channel. You're good.
Say what?
You're on your normal channel. So you're good.
Meaning what, son? You're watching my live stream right now?
Yeah.
Sweet.
It's on RyanReardon71.
Okay.
So it's in the right channel.
Well, but what about these folks?
I don't see them on this channel.
Yeah. See, we're in the wrong place. We are in the wrong place. Where am I? Where is this? Waiting for Ryan Reardon. TR7.
You might have 2 streams going at once. I had 2 notifications for the stream on my lock screen. So
Yeah.
wait, I'm out of it. Okay, now I'm in the one. Now I'm in this one.
Yeah. So how do you get there? How do I get there?
This one's not live.
Right. So how do I get people to my live dashboard?
I think you need to exit out of that stream and go to the other one. So right now you're in the wrong stream.
How does that work? How do I get people over here?
You could send them a link.
What's the link? Here?
Yeah.
This stream? Son of a bitch. Hey, you're in this one right now?
Yes.
Hey, say this is James's. This is Mr. Reardon's son, James. We're trying to figure out how to get. Okay, thank you. So David should be hooking me up.
Wait, do I need to still say that?
Yeah. Oh, I know what this is. I know the deal. Hey, Allison. Hey, Allison, if you can hear me. You might also mention that Mr. Kanufki is going to get everybody over, send everybody this link.
Wait. You just type it in. You realize your channel is being edited?
Yeah.
You're asking me to put the right link, so.
Oh, yeah? Do you know the link?
No.
How did I screw that up? I remember.
Somebody just told me you got this.
Hey, just checking the volume. We're still streaming. I wish I could find it on the internet.
Right, when you have the right way to tell me so I can type it in.
Hey, Sydney, can you hear me?
I'm missing lunch for this. Come on.
Excellent. My son is missing lunch. He's my tech support.
Yeah. Yes,
Oh, you're missing lunch. Dar, we'll hook you up. Miss Patel will hook you up, man. You're all good. Hey, David, if you could send... Well, Colin, the problem is that I have an updated link, and the published link is old. I neglected to update it. Let me know if you can hear that, Mr. Davis. All right. We'll know when people are... Okay, hey, Colin. Let's just test the system here, buddy. What's red and smells like blue paint? Thanks, David. So what are you watching right now?
I'm watching the wrong one.
You're watching the wrong one. That's exciting.
I'm gonna like once we have the link.
Cool. Thanks, Sydney.
Once we have the link to get them to
Hey, so if anybody that's out there can beat Colin to the punchline, what's red and smells like blue paint? Yes, Colin, it is red paint. Nicely done, son. We're waiting on other folks to join us since I was a technical newbie and neglected to update the link. Biology, I know. Updating links on YouTube, not so much. I'm learning. That's the whole reason for doing this. I'm learning. 45 people waiting. We have 45 very upset people on the other link.
And I think my dad apologizes.
Yes. Hey, Mr. Kanufki, if you could type in the published link here, I'll have my son pop it over to the other chat. I'll try and type that too.
I said my dad apologizes for sucking at technology.
You did not say that. Apologies. Guess my son is going to run for president in 30 years. Thank you, Sydney. For those of y'all that are listening, you know that yesterday, well, there's only like 4 of you, there were storms in Kansas. Today we've got storms coming in Alabama, so we're going to try and get this done before the tornadoes come. Hey, James, tell Jill that, all right, thank you, David. 21, we have 45 41 on the other stream.
Wait, what do I tell Jill?
Tell Jill we're going to touch on the Krebs cycle and the Calvin cycle. Okay. David Kanufke, the master of ceremonies, is saying, let's go for it. So let's go for it. Hopefully folks will jump on over.
How do I spell this?
You spell them right there. So I've got my son communicating with the lost ones over there. I'm going to move my camera so you guys can see me. Good afternoon. It should be 1:11 Eastern Daylight Time. Down here in Alabama, it is 12:11. We are awaiting storms. My name is Ryan Reardon. Mr. Kanufke was kind enough to invite me to lead an online session reviewing, now not teaching you guys, but reviewing some complex topics in AP biology. It's going to be about an hour and a half, so settle in. It's going to be a lot of talking. If you've got questions, please send them over to the chat. Miss Allison and other folks will be taking care of that chat and feeding me information. Sydney Bergman, a good friend of mine from the DC area, is also going to be moderating. She is online as well. And again, nice to see you guys. Kind of thinking about the overview, you can see it here below the title. When we started looking at, we sent out a survey for complex concepts in AP Bio, and the thing which kept coming up, and something that's similar to my kids as well, is the cell communication piece. That's always a challenge, and so we want to look at some of the nuts and bolts of cell communication. There were a few line items on gene regulation, which I will briefly go over. Specifically, we'll look at the difference between operons and which is a prokaryotic mode of gene regulation. And then we'll look just for a quick second at some eukaryotic modes of gene regulation. And I'll also kind of talk about, from my perspective, what to expect on the AP exam in terms of eukaryotic gene regulation. And again, even though I know that my friend Bob Kuhn spent a good bit of time on energetics, that's a big idea too, there was still a call for a discussion of chemiosmosis. So I'm going to give you my take on that as well. And then I've also got up here grab bags. So if you've got burning questions, throw them in the stream and we will try and handle those. Now, I'm going to tell you guys something I tell my students. I do not have a cornerstone on wisdom. I do not have a monopoly on knowledge. There are hundreds, if not thousands, of excellent AP Bio teachers out there. If I say something that contradicts your teacher, it's just my perspective. Again, I may even be out of date on some of this stuff. I hope that I'm dead on. And if something comes up, just engage with your instructor. The adult that works with you on a daily basis, loves you, cares about you, and wants you to be successful a week from tomorrow. So with that said, let's get started looking a little bit at cell communication. Let me get this thing going here.
To get these people.
Okay. Well, they'll come on in. They'll get there. Let's get going. The boss says, let's go. I say, let's go. So I'm going to my next slide. So here is a...
Did they have a link for it?
I think they've got it. It's published somewhere. This is a slide that most of you guys should be familiar with. This is coming out of Campbell's Biology, 7th edition. And here what we're looking at is your simple cell signaling pathway. The keys to a cell signaling pathway are as follows. You've got a receptor, which you see over there on the left in purple. Quite often you'll see the purple color indicates a protein in any Pearson print, whether it's biology or focus on biology. And then you've got this orange molecule that looks like a teardrop. The fancy word for that is ligand. Lig, like ligament, means connector. So it's a molecule, always a small molecule, that connects with that protein and causes a shape change. Now, the big part, which we'll get into in a minute, is the middle right here, called the transduction. That is the relay of signals, not too dissimilar from a bucket brigade, where one protein activates another, which activates another, which activates another. This one's live now. Hopefully folks have got it. And then ultimately, at the end of signals, okay, let's cut that off.
Yeah,
okay, thanks. Excellent. We are in business. Y'all can see me. Ultimately, the end of the signal transduction pathway is going to be some type of cellular response. And we're going to look at several of those responses today. Looks like we are on delay. Yes. So, hey, guys, and again, if you have questions, ask Sydney. What is going on with that? Are we frozen? Keep that open. Okay. Wow.
everybody to go to your channel and click on the actual pipeline.
Okay, thanks, man. We are dead in the water, but I'm going to keep on going. We may get some... Okay, you can still hear me, but you can't see me. Thanks, Sydney. Thank you very much. So if you're looking at this slide, ultimately, you've got that protein connecting with a ligand. That sets off just a cascade of signals within the cell, small molecules interacting with each other, and ultimately you get a cellular response. I think, and that's pretty straightforward to me, I think the bugaboo for folks is the actual phosphorylation cascade. And so let's look at that next, you guys. Let's take a look at that. And so what I've got is, this is not very helpful for y'all, but what I've got is a picture of a phosphorylation cascade. So since you're looking at those, James, what are you seeing, buddy? Just a
I'm going to give, so the link's up there, I'm going to give them the link.
Are you frozen? Oh, you're still doing the link. Anywho, if we consider these relay molecules, what we need to recognize is that they're sort of, not their priority, but their default setting, for lack of a better term. Their default setting is off. Essentially, when you think about the way a cell works, Swathy, virtually all systems use a phosphorylation cascade, buddy. It's just going to be, it tends to show up in the immune response as well as the endocrine response as well. Okay, cool. You see the new slide. Awesome. Thanks, David. So check this out. Let's just close that down, man. This is bugging me.
I need the link.
Okay. Hi. So when we look at these small molecules, what we're looking at is they are getting tag teamed by a pair of other proteins or enzymes. One is called a kinase and one is called a phosphatase. Now, the kinase is an important molecule in AP Bio. It's an important molecule in all of our cells. The thing that we need to recognize is that kinases activate other molecules. The way they do that is by phosphorylating other molecules. And so we get to play a little bit of a game. If you want to make something happen in a biological system, you need to destabilize it. That, my friends, is what a phosphate group does. So when we think about the role of ATP in cells, yes, it's the energy currency of cells, but the question becomes, what makes it the energy currency of cells? Well, bottom line, not to get make too fine a point, but it destabilizes other molecules. It makes things happen. So now I want you guys to be kinases. Or I want you, no, let me change the groove. I want you to be phosphorylated. Okay, so imagine this. You are a molecule inside of a signal transduction pathway, and a kinase just came up to you and phosphorylated you, which means you just obtained a phosphate group. Now, if you're sitting down, you're pretty stable. So now that you've been phosphorylated, I want you to stand up. When you stand up, you are less stable than you were before. Now, let's imagine that you're going to get phosphorylated again. Pick up 1 foot. And if you notice, I'm picking up 1 foot, and you can see me start to wobble. I'm becoming less stable. Now, to carry the analogy further, close your eyes. And if you put down your arms, and I just fell over. So that's a game that my students and I play when we look at, we think about phosphorylation. Ultimately, these molecules in the signal transduction pathway, their kind of steady state is off, but then they get turned on when they are signaled by the extracellular environment. Now, think about this for just a quick second. If they stay on all the time, the cell's going to go haywire. So in addition to having a kinase, each molecule in the signal transduction pathway also has a phosphatase. That's an enzyme which removes that phosphate group. Thank you, Sydney. I'm going, baby. I'm going. That phosphatase pulls that phosphate group off and returns that molecule back to its default setting, which is off. So, and again, I realize that's a really simple analogy, but ultimately what you've got here is this conversion or bucket brigade of one molecule getting activated. That leads to the activation of another molecule, which leads to the activation of another molecule, which leads to an activation of another molecule, and then so on until ultimately you get a cellular response, which you can see down here at the bottom of this slide. And notice again that this active molecule is phosphorylated. Now, and again, just one more, not to belabor the point too much, but you don't want those proteins always activated. They get turned on and then they get turned off, and that's essentially how a signal transduction pathway works. We're going to look at several applications of that fairly complicated idea. Now, I would say this in terms of test prep: if there is a short free response or maybe even a longer form free response and you're asked to describe a signal transduction pathway, I would definitely start with that connection between the ligand, which you're seeing at the top of this slide, and the receptor. That receptor is going to change shape. It's essentially going to be destabilized. That's going to kickstart this phosphorylation cascade. I would describe that activation and deactivation of particular proteins, and absolutely positively I would describe some cellular response. We're going to look at a couple. You don't have to use mine on the exam, obviously, but you want to use an example that makes sense to you, because you definitely want to write from the heart when you're writing those FRQs. So I'm going to switch slides real quick, my friends. That is the previous slide. That's not very helpful. There's the start. I'm that guy that can't figure stuff out. All right, we saw that. Now we should be good to go. Now, a lot of folks were asking about second messengers on the on our little kind of pre-talk survey. So, what's a second messenger? I know you guys can all read, but a second messenger is a small water-soluble molecule that diffuses quickly throughout the cytoplasm. In a sense, it's a PA system for a signal. So check this out. Here in this particular example, we've got a G-linked receptor. We call it a G protein because it's shaped like because it's shaped like a G. I should know that, but I don't. I apologize. Here we have our ligand, again, this orange teardrop. It's the first messenger. What's cool about this particular ligand is that it is not going to enter the cell. Notice that it's going to bind with this G protein, and it's going to cause a change in that G protein. What happens is, thanks, Bob. You posted the URL to Facebook. Thank you very much. Yes, because it's thanks, Sydney. We call it a G protein because it's associated with ATP's cousin, guanine triphosphate. Or this is what we've got here. Actually, I apologize. It's a G protein linked receptor. It picks up the ligand. It causes that G protein to be destabilized, and then that G protein communicates with another molecule. So essentially, you've already got 3 steps before anything has happened. Now, so what? Here's the so what. When that adenylene cyclase, or this cyclase, that third molecule in the chain, is activated, it sends out this second messenger. Often, it's cyclic AMP, or what I tell my kids, we call it CAMP. It's cyclic adenomonophosphate. It's a cousin to ADP and ATP. But what happens, that cyclic AMP, it can spread like wildfire through the cell and activate multiple proteins at the same time, so that you get a faster response. Essentially, this molecule outside the cell is calling on the G protein link receptor to make something happen, but then it disengages from that receptor and moves on to another cell. So ultimately, that messenger can activate lots of cells. It doesn't have to spend the time telling the cell what to do. It just tells the cell to do something. And it's the second messengers that send the signal of do something. So if I remember my history right, 1 if by land, 2 if by sea. Paul Revere sees the lamp in the tower. He jumps on his horse, as legend has it, and he rides through Boston telling folks the British are coming. And then he doesn't stop and tell people what to do. They know what to do. They grab their guns. They go to the community green, and the rest is history. Here we are in 2017? You could think of Paul Revere as the original ligand, the messenger, and then the cyclic AMP, or those kind of courtyard and township organizers. I know it's a cheesy analogy. I just thought of it. I should have probably shut up. But that's second messenger, you guys. A small molecule that diffuses through the cytoplasm because it's water soluble and amplifies a signal, so that the signal can be used in other places. I hope that makes sense. If it doesn't, yeah, absolutely. We're definitely going to go longer. Now, if you want to think about how this works in a real situation. Let's take a closer look at some specific, at least I would say general examples. How about that? Going to next slide. So here, I've got a pair of slides. I'm going to move my face over. Shoot. Hang on, guys. Move that way down. My apologies. Go away. All right, got my laptop camera. Let me move over here. This is trial by fire. All right. Let's pay no attention to this image behind my not so pretty face. What we're looking at here is a ligand like this is basically the same situation we've looked at the last few times. Here, our ligand molecule is binding to a membrane-bound receptor, and that's because this particular ligand or signal molecule is polar. It's water-soluble. It's not going to diffuse through the cell membrane. It's going to attach to the receptor, and then as that receptor has that shape change, it's going to set off the signal transduction cascade that we discussed a few moments ago. What's interesting about this particular slide is it actually gives you a feel for so what happens next. In a sense, in this particular cartoon, I should say, what's happening is at the end of that signal transduction cascade, you've got a transcription factor, simply a protein which actually can bind to a promoter region of some DNA and activate that gene. Here, what we're seeing in the nucleus of this cartoon cell is that we're seeing an inactive transcription factor, and here we're seeing it activated. And when it's activated, check it out. There's your phosphate group. It's destabilized that protein, so it's going to bind specifically to a certain region of DNA. It's in a sense going to turn that gene on, open it up for expression, more specifically transcription, and you're going to get a messenger RNA. I am not going to spend a lot of time talking about the nuts and bolts of transcription and translation, but ultimately what you're looking at there on the left is a change in gene expression due to some extracellular ligand. Now, let's move over here. All right. Second verse is pretty much the same as the first, but here our ligand is a hormone, testosterone. It's a lipid. It is nonpolar. It is not soluble in water, but can ease on through the cell membrane. As such, its receptor is not embedded in the membrane, but it's actually inside the cytosol. And here what we're seeing is we've got a receptor protein binding to this hormone, testosterone. Now, just like in the slide we saw a moment ago, or the image we saw on the left, that receptor migrates into the nuclear envelope. It is unstable. It binds with a specific region of DNA and leads to the transcription of a particular RNA transcript, which is then edited. That RNA transcript leaves the nucleus for the chaos of the cytosol, and it is ultimately transcribed into protein. So if this were testosterone, that protein might be a growth factor. It might be more follicle, more cells in the epidermis, which are going to lead to armpit hair. I mean, I don't want to try and keep this PG, but ultimately, if you got testosterone coursing through your blood, that's going to call many cells into action, and it's going to lead to secondary sexual characteristics of males. Now, because everybody likes pictures of dogs, puppy dogs in particular, I thought that James, be quiet, please. I thought that I would show some pictures of my dog who would be dealing with all these growth factors. Now, it was going to look a lot cooler when I had this as a keynote presentation, and dang it, that's the previous slide. I'm going to figure this out. Next slide, Reardon. So you're just going to see a bunch of pictures of my dog piled on top of each other, but she's pretty darn cute anyway. So here she is about a week and a half ago, and then we're going to go back in time. We can see what she looked like when she was younger. Oh, there she is about 2 weeks ago. there she was before all these growth factors really kicked in. She's a cute little puppy hanging out with her handkerchief, or my wife calls a scarf. And then here we are about a week and a half after we got her from the pound. We realized that she had a broken leg, but because she was stuck in a crate, we didn't know it. So here's cute little Mary Bailey as a beautiful little puppy dog with her cast on, and it's a fun story of cell signaling, because now she is a healthy, happy, slobbery, beautiful, hairy black lab mutt, and we love her. And the reason that she's growing so much is because of all those ligands that are coursing through her bloodstream, connecting with her cells, and ultimately causing her to grow. Now, let's move on. What about short-term changes in cells caused by cell signaling? I want to look at one of my favorite examples, which is cardiovascular physiology. So we'll move on to the next slide. And here what we're looking at is, let's just take a look at this particular slide. What we've got here on the left-hand side, kind of moving in storyboard, 1, 2, 3, is we're looking at how cell signaling might work in the central nervous system. And again, I realize this is a super broad stroke. It's a quick look at how this works. But ultimately, here our ligand is a freaking neurotransmitter. That's all it is. This receptor, you know it from your discussions of the central nervous system. It's a ligand-gated ion channel. So that's quite a big change for this particular protein. When the neurotransmitter, which is specific to this neurotransmitter receptor, it's a receptor just like any of the other ones that we've looked at today, when that ligand binds, that channel opens up. And when that channel opens up, ions that are outside the cell have the opportunity to diffuse inside the cell. You can imagine, so dig this. If this ligand right here on the top left, this orange teardrop shape, if that's an excitatory neurotransmitter, let's just say epinephrine, it's going to bind with a sodium channel, a ligand-gated ion channel. It's going to open up, and it's going to allow sodium to diffuse into the cell. That's going to cause a depolarization of said cell and increase the chances that that cell will actually generate an action potential. When that ligand goes away, when it's absorbed by a neighboring synapse, a neighboring cell, a presynaptic terminal, if you will. If two neurons are talking to each other, we'll do it that way, right? So if this is the presynaptic terminal, releases the neurotransmitter. Here in my left hand, your right is my postsynaptic terminal. It's going to bind to the receptor, and then, and this is going to be by magic, the presynaptic terminal is going to suck up that neurotransmitter again. And that's an important point. You don't want those ligands just hanging around between cells in the central nervous system, or even the peripheral nervous system for that matter, because it's going to cause changes in neurons and excitatory potentials, action potentials that aren't needed, right? In a sense, hallucinations, if you will, and if you are schizophrenic, it's an increased excitation towards of nerve cells, for lack of a better term. Essentially, individuals are seeing things that aren't there because certain nerves are excited. They're hearing voices that aren't there because certain nerves are excited. On a more mundane note, you've likely heard of things like Paxil and Zoloft and other antidepressive drugs or anti-anxiety drugs. They're called SSRIs, selective serotonin reuptake inhibitors. Serotonin is a neurotransmitter which opens up certain, actually what it does is it's going to bind probably with potassium channels. Potassium diffuses out of the cell. That actually lowers the resting potential of particular cells and kind of slows them down and chills them out. When cells are bathed in serotonin, you feel relaxed. Well, that serotonin is actually taken up, and then you don't feel as relaxed. The drugs like Paxil and Zoloft that I mentioned, these SSRIs, reuptake inhibitors, what they do is they allow those neurotransmitters, which lead to a calming effect, to hang around inside the synaptic gap and bathe cells, hopefully reducing anxiety, relieving depression, and allowing folks to work more efficiently. Now, that's all in the central nervous system. Let's move over and take a look at the cardiovascular system. And so here, I want to spend just a quick second looking at maybe the specifics of how the cell signaling works inside the cardiovascular system. So I need to move my screencast over so I can see what I'm talking about here. I was talking about this with my students just the other day. Actually, with the students I'll get to teach next year. So I teach a second-year HL biology class, and so unfortunately I don't get to teach a lot of the cell signaling stuff anymore because it happens during year one with my brilliant colleague, Ms. Breland. But ultimately, what's cool about the heart is that it has this beautiful little patch of cells inside the little top notch of the it's actually in the sinus of the atrium, a gap inside the atrium, so we call it the SA node, and this is hanging out in the right atrium of our heart. That's our pacemaker, and you've likely heard of pacemakers before. You may have a relative that has had a pacemaker put in. When you have a pacemaker put in, essentially you've got a tiny machine and computer that is sending a signal to the heart. I don't want to get too deep into it, but ultimately it's zapping the heart with just a little bit of electricity to get it to beat regularly. What's cool for healthy individuals is their hearts are going to beat regularly anyway, because this little patch of cells, the SA node, actually spontaneously depolarizes. It has the ability to generate action potentials all on its own. And so you have a resting heart rate. Maybe it's 60 beats per minute, maybe it's 70 beats a minute. And there you go. Now, and I think I've mentioned that already. We've got the SA node depolarizing, you've got just a resting heart rate. Now, let's imagine this. You lied to your parents. And they caught you. He caught you bold face in that lie. You know you're busted. Well, you're going to have a response to that. It's quite likely that you might start to sweat. Your heart's going to start to beat a lot faster and a lot stronger. Why is that? That's because when you know you're busted, or you get essentially, whenever your sympathetic nervous system gets energized, there is a nerve called the vagus nerve, which is going to dump epinephrine on the SA node. You can see the text here on the right-hand side of this slide. What's cool is that the SA node, let me say this, there are receptors for epinephrine in the sinoatrial node. When they receive that epinephrine, what's going to happen is that those voltage-gated ion channels are going to open up, more specifically sodium channels. That's going to allow sodium to diffuse into the pacemaker cells. It's going to get them closer to it's going to get them further away from minus 70, closer to the threshold potential, and boom, the SA node is going to start to depolarize faster and more often, so more frequently. That's the increased heart rate. What's also cool about, and you'll see that here, I've got the notes and now I'm not even using them. The receptors for epinephrine lead to an increase in SA node permeability to sodium. That, again, just to be very specific, that increases the likelihood of depolarization, meaning that the heart's more likely to beat faster. What's also cool is that epinephrine also alters the cardiac myocytes, that is the muscles that line the interior walls and the exterior walls of the atria and ventricles. That's going to lead to an increased strength of contraction. So not only is your heart beating faster, but it's also, the stroke volume is increasing as well. And that's why you get flushed in the face and why you start to sweat a little bit, because a lot of blood is going to your periphery, getting you ready for that fight or flight syndrome that we're so used to. And again, I realize that's a lot of information on cell signaling, but that's what the people wanted, and so I wanted to make that. I think next we're going to look at cell signaling in plants, because that was also something that came up, and I want to spend just a quick second there as well. Hey James, would you get me a cup of water, please? If you get a second. Thank you, sir. I need a cup of water, buddy, if you don't mind. Thank you very much. Oh, no, I lied. We're not on to plants yet. We're on to the immune system. This seemed to be the most popular topic. So let's talk this out for just a quick second. This is not, hey guys, let me be as clear as possible. This is not going to be an hour and a half long talk on the immune system. I'm going to give you about 12, 15, 18 minutes on kind of the overview. And I realize that it's complex, but it's just not that complex. The thing I want you guys, it's not that complex in terms of the level that you need to understand it for the AP Bio exam. And really our hope is for those of us that teach this stuff is to sort of open up doorways, get you thinking about how this stuff works, maybe inspire you to take another class when you get onto college. And again, not everybody has to be an immunologist or working in a STEM field, but ideally you know more about how the world works and you want to learn more, because we clearly can't tell you everything inside of one class. Okay, hey Sydney, where are we in the lag? How far back did we get? Because right now I'm looking at slides on the immune system. I've got a picture of blood vessels and a picture of a lymph node. I'm curious about what y'all see. You don't know. Okay. You don't know what we see? What are you looking at right now? Zone of lag. Okay. Sweet. Good. Awesome. Well, here you go. One of the things that kept coming up in the stream of questions was this concept of humoral versus cell-mediated immunity. And I want to talk about that for just a quick minute, but I wanted to put that in the context of the cardiovascular system. When we think about humoral immunity, let me move this camera over. When we think about humoral immunity, we're really thinking about B cells, and I would just make the argument about the antibodies that the B cells secrete. Here what we see are these purple Y-shaped things in this cartoon are models of antibodies, proteins that have a specific shape that, as all of you probably know, are specific to particular antigens, more specifically specific to particular epitopes, that is, shapes, recognition sites on antigens. Okay, I heard that. It sounds like, yeah, humoral is fluid like antibodies. Okay, it sounds like we're all back on the same page. And I really appreciate you guys sticking with me through the glitches. Now, the reason I have a picture of the bloodstream here is because that humoral immunity, that's going to occur within the capillaries. This is proteins that are inside the blood that are being carried through your bloodstream that are essentially helping your body mop up any foreign agent. So that's when we think humoral immunity, we want to think B cells, and particularly we want to think antibodies. And again, I recognize that that's kind of an oversimplification, but that's a good way to kind of get your head around it. Now, when we think about cell-to-cell mediated immunity, here we want to dive a little bit deeper inside of the lymph nodes. So if I teach out at Campbell and focus in biology, so we have this awesome picture of this guy who's transparent, all his lymph nodes are out there, and he just looks so relaxed. I wish I could be as relaxed as that guy. But if we were to, if we were to pluck out one of the lymph nodes, let's say it's a lymph node near his mandible, or maybe it's an axillary lymph node, it's under his armpit there. What we would see is this beautiful kind of honeycombed webbing of blood vessels and also lymphatic vessels, these blind-ended tubes that pick up fluid from the blood, clean it up, and then return it back to the heart. And so this is in fact, these two pictures above my head are actually pretty darn useful when you think about how cardiovascular physiology and basic immunology works. And you guys know this, but let me just explain it anyway. When your heart beats, it puts pressure on the fluid inside your arteries and your veins as well, but mostly on your most of that pressure is going to be on the arterial end of the cardiovascular system. Fluid leaks out of the capillaries. It just does. That fluid then, which we call blood or plasma when it's inside the capillaries, we call it interstitial fluid when it's between the capillaries and the cells. And then when that stuff gets collected by the lymphatic system, we call it lymph. So if we're talking about the fluid constituents of the blood, or we're talking about interstitial fluid, or we're talking about lymph, we're really talking about the same thing. It's basically what's in the blood that is not cellular. Now, when that stuff gets inside the lymphatic system, and particularly these lymph nodes, that's when your T cells can really go to town. And we're going to talk a little bit more about that. But hanging out inside the lymph nodes are all these beautiful B cells and T cells that essentially find specific regions on antigens and recruit other cells to attack those invaders. And I know it sounds like war, and I said beautiful, so I really feel silly because I feel like That's something our current administration, head of our current federal administration might say, so I'm trying to avoid that. I also want to try to avoid being political, but sometimes I just can't help it. But it's those B cells and T cells that are hanging out and getting super active inside your lymph nodes, and that's why your lymph nodes swell, your glands swell when you're sick. It's because you've recruited more tissue. The fluid is filling up, getting inside those lymph nodes faster than it can get out. It's one of the things. That we. I'm reading messages and trying to talk at the same time. I'm a male. I don't multitask. I apologize. The lymph nodes are honeycomb shaped, and essentially they have evolved to take in more fluid that they can let go. That slows down the fluid. That allows the B cells and the T cells to do their thing. But your lymph nodes swell and they get hot they get warm, I should say, when you're sick, because not only do you have more fluid and more cellular activity there, all that cellular activity tends to increase the localized temperature at your neck and under your armpits, and sometimes you can feel them in your groin and behind your legs as well. Wherever you've got lymph nodes, you're going to have B cells and T cells. Okay, and now that I really spun out of control on that one, let's take a look at some more specifics, shall we? Next slide. Yeah, you guys are going to be here till next Monday at this rate, so just hang with me. Next slide. All right. Now, you've probably seen these in your books before, so now I'm going to do a little hoochie coo here. Shoop. Shoop. All right. Bottom line, we're thinking about acquired immunity here on the left.
This is branded in India.
On the left, we've got the humoral immune response, what's happening in the blood. On the right, in this beautiful kind of seafoam green, we've got the cell-mediated response. Ultimately, you've got exposure to an antigen. We're going to look at these dendritic cells. That there will be cells within the bloodstream that can grab onto those antigens, pull them apart, and then present them to our favorite cell, the kind of the mediator of the immune system, the helper T cell. As many of you guys know, that's the cell which also is host to human immunodeficiency virus. And when your helper T cells, specifically your CD4 helper T cells, go down in concentration, that's gonna cause immunodeficiency, because these helper T cells, particularly the CD4s, they're the guys that are, or I shouldn't say guys, they are the cells which are activating all the other cells. So in a sense, it's getting rid of the commander, the captain, quarterback, whatever you want to say, whatever analogy you choose to use. This is the boss, man. And when you take out the boss, these other cells don't know how to act, at least very well, and so you have immunodeficiency. Now, let's see what I got here. Let's see if I can't pull some of these images back together. Now, yeah, let's just take it for what it is. Let's look over here. Let's look at the left side, the humoral immunity. When helper T cells are activated by dendritic cells, and we're going to look at how that happens in just a moment. They're going to activate B cells. These B cells are going to go into a rapid cell cycle. They're going to give rise to two sets of cells, plasma cells and memory B cells. So imagine this. There is a ligand secreted by a helper T cell, which changes the cell cycle within the B cells. It's really fascinating stuff. And again, the details of it are well beyond the scope of AP biology. You've got these plasma cells, and you'll notice even in this cartoon, you've got lots of endoplasmic reticulum. That's going to indicate that these cells are going to be protein factors. I should say rough ERs. They're protein factors. They're going to be making and secreting antibodies and secreting those antibodies into the bloodstream, hence the humoral response. What's really slick is some of those B cells aren't active now. We call those memory cells. So they've been activated at the same time. They have been trained, for lack of a better term, to recognize a particular antigen, and then they just kind of hang around in the bloodstream waiting for being available in case that same antigen, in case the body comes into contact with that same antigen. This is essentially why you get vaccinated. A vaccine is going to be epitopes of particular antigens, your body recognizes that as foreign material, and you are going to make B cells, which will then give rise to plasma cells and memory cells. The plasma cells will just take out of whatever little bit of heat-killed virus you have, and then your memory cells will hang out ready to pounce on that antigen if it ever presents itself again. It's likely, I know that I had chickenpox when I was a kid, and it's likely that my moderators had chickenpox as well. It's likely if you're watching this screencast, you've never had chickenpox because you were vaccinated for it. You're figure out how to develop the vaccine. We figure why make kids get sick and itchy and have fevers and have chickenpox. We can present them with chickenpox virus. They can develop an immunity to it, and it's one less thing they and their parents have to worry about. It makes perfect sense. So remember, kids, get vaccinated. It makes good sense. Now, on the other hand, let me see where I am let's see where my face is. Let's move us over to the cell-mediated stuff. Again, the second verse is pretty similar to the first. Helper T cells are going to send out signals. Those signals, which we'll name they're called cytokines. We'll look at them in just a second. They're going to recruit what we call cytotoxic T cells, killer T cells. These helper T cells direct the helper T cells direct the killer T cells to go and attack those antigens, to saddle up next to them, to secrete proteins out of their cells that essentially increase their permeability, cause them to shrivel up, kill them off. Much like with our B cells, we've got these activated cytotoxic T cells which do the removal of the antigen, and then at the same time we've got clones of these active cells that are just going to hang out as memory cells ready for that antigen to present itself again. So in case you did ever get chickenpox, your body would not only have antibodies to attach to that chickenpox virus and slow it down, you'd have cytotoxic T cells that could go and attack that virus, break it down as well. And again, I realize you may have an instructor that is much more well-versed in immunology than I am. I learned this essentially as a teacher. If you've got somebody, in my background is in ecology and molecular genetics, but if you've got somebody with a stronger immunology background, ask your instructor about this stuff. And hopefully they'll tell you, look, we're just looking at the basics here, the basics of the complicated processes, not totally getting into the weeds. But speaking of the weeds, I do want to get in there for just a quick second. Here, there's something that's almost guaranteed to come up on the exam, at least in a prompt, and that's this idea of multi-histone compatibility complexes, or MHC1 and MHC2. And it's one of those things where if you're not prepared for it, you may not be successful at on that prompt just because you kind of get freaked out. Now here's the way I teach it to my kids. All nucleated cells in your body have what we call MHC1, or multi-histone compatibility complex 1. These are proteins which can bind to epitopes and then present those to killer T cells, or what this cartoon is calling cytotoxic T cells. In a sense, when a cell is attacked by an antigen, it's going to die. And so let's say that the cell is attacked by a virus, the virus takes over the cell, and that cell is dying. But as it's dying, its last dying wish is to grab pieces of that virus, and as a killer T cell comes there and says, "It was that virus," and then those killer T cells know to go attack that virus. They've been activated by this cell with MHC1. On the other hand, we have very specific cells called macrophages, and then we've also got dendritic cells as well that have what we call MHC1. These cells' job is not to get attacked by a virus and essentially give their last dying message, "Oh, it was the virus." They go out and find the virus or the bacterium or whatever it is that happens to be infecting your body at the time. They take that thing, they rip it apart into its constituent components, and then they feed those constituent components to an MHC2. So they're like, "Hey man, we got an attacker here, no problem. Let's go get it. Let's rip its arms and legs off. Let's present those arms and legs to a helper T cell. That helper T cell says, 'Oh, this is the guy I got to go get,' or this is the antigen that I'm looking for. These are the epitopes that are showing up on that antigen. Now that I know what I'm looking for, I'm going to go direct killer T cells and B cells to killer T cells to go and attack that antigen, B cells to make antibodies to that antigen to make the T cells' job easier. So in a sense, these guys are cells with MHC1 are victims that are communicating with a cytotoxic T cell. Dendritic cells and other cells with MHC2 or multihistone compatibility complex 2, a word only a biologist could love. They have evolved to attack antigens. They're not the victims, but they are the predators, for lack of a better term. They attack those antigens, they tear them to pieces, they activate their MHC2, these protein bridges, and then communicate with helper T cells and say, hey, this is the problem, go take care of it. Amazing things, these dendritic cells, just beautiful, beautiful cells. All right, looking forward, moving on. La la. Next slide. Yeah, here, okay, just some more details on this idea of dendritic cells. La la. See my forehead there. So here we've got a dendritic cell. It's attacking an antigen. Here it's a bacteria with triangles on its cell wall. What's happening here is the thing that we really want to focus in on. Let's see if I can actually make this a little larger. I can. It's an emphasis on the chemicals. These little dots here. These dots, they take us back to our first couple of conversations about ligands. We call these chemicals secreted here by the dendritic cell, and then later secreted by the helper T cell. These ligands are what we call cytokines. Pretty straightforward term. It's a chemical that excites or activates other cells. You may see this in a prompt. We call those particular cytokines, we call them interleukins. Interleukins, I mean, break the word down, it means between white blood cells. So if a macrophage and other dendritic cells is a white blood cell, if a T cell is a white blood cell, and B cells are white blood cells, and there's going to be communication between them. We're going to call those specific modes of communication interleukins. This is communication between white blood cells. I hope that makes sense. And if we carry this analogy forward, we kind of look at what's the end result of the killer T cells and the B cells. This is kind of the bottom line, you guys. The cytotoxic or killer T cells are going to mediate that cell-to-cell response, while the antibodies, which are secreted within the bloodstream, those guys are going to be that's going to handle the humoral immunity. And again, I hope that makes sense. Haley, where can we have access to those slides? I will make sure that I turn them into a PDF, and I will post them on. I'll email them to David, and maybe he could post them out there to the world. We'll figure that out by the end of the day. Okay. MHC1 and MHC2, Reshma, essentially it's just different proteins that you're going to find in different cells. That MHC1 is going to be found in all nucleated cells, while the MHC2 are specific to macrophages and dendritic cells. In regards to the difference, it's likely different sequences of different structures because they're coming from different genes. All right, moving on. La, la. Next slide. I think we can leave the immune system and move on to... Oh, if Colin and Elijah are still watching, they know that if there's one thing I love, it's plants. Come back to you. Plants, and more plants. Oh, plants. How do plants know things? They don't know anything. Plants aren't sentient, yet plants can respond to daily and seasonal and annual changes in the environment. I'm doing Trump hands now. I gotta stop that. I'm sorry, you guys. They're amazing. Plants are awesome. How do they sense changes in the environment? Well, they do it lots of ways. But one of the coolest ways that they do it is by circadian rhythms. They can tell time. They know what season it is based on day length. How the heck do they do that? They do it with these really cool proteins called phytochromes. So let me just pull this up. I'm going to get rid of myself here if I can. Boop. Yes, it's probably better for everybody. Now, here we are looking at a phytochrome. It looks like bunny ears or a couple of bowling balls. Teenage boys, you might think it looks like something else, but mind your own business. Now, here's what you need to know. These phytochromes exist in two isomers, that is, two different shapes. We have PR, which stands for phytochrome red, and we have PFR, which stands for phytochrome far red. And so let's talk about what the heck we mean by that. When we say phytochrome R, that's a protein which is going to be stimulated by red light. And when we think about this red, we're talking about stop sign red, we're talking about candy apple red, somewhere around 680 to 700 nanometers. Now, that candy apple red light is going to be a part of the visible spectrum. Obviously, we don't see it because we see white light, a mix of all the spectra. But when you see somebody wearing a bright red sweatshirt or you see a stop sign, you're seeing a reflection of that red light because all the other wavelengths are being absorbed. But what's cool is there's also this kind of dusky dark red, almost kind of a dusky red maroon, and biologists call that far red light. This is a longer wavelength. Look, you're looking at about 710 to 730 nanometers, and it's got slightly lower energy. That far red light is usually going to be available to plants when the red light is unavailable. So think about, and this is a complex idea, and just hear me out. Plants that are exposed to the full visible spectrum are bathed in red light. That being bathed in red light is going to lead to several different responses. We'll look at one in just a second, but things like controlling when flowers are blooming, whether you have a short day plant or a long day plant, that's going to be set up by the phytochromes. Essentially, the plants are telling how long the days are based on the amount of full visible light that they receive. Flowers that bloom in the winter, like a camellia down here in the southeast, are what we would call short day or long night plants. And then plants that are going to bloom in the summer, like the echinacea down here that are about to bloom, or the hydrangeas that are just starting to bloom in my backyard, those are what we might call longer day plants. They essentially they need to be bathed in more red light for longer set periods of time before those cells will initiate flowering. Now, blah, blah, blah. What's cool, and this is what I think is cool. I'm going to bring myself back in here because I just love talking about this stuff. Am I back on? Uh-oh. Hopefully, there I am. Hopefully I'll show up. Move that up, laptop camera. Up. Hello. All right. Technology. Now, hey kids, I want you to imagine a forest. And you're walking through the forest. What's the light like? Hopefully you're seeing this sort of beautiful golden hue. Not really saturated colors, but sort of a dampening color. You've got this again, I'm in the southeast, so I'm used to walking around in forests. If you're in a more arid climate, you might not be used to being in dense forests. But when you're under that forest canopy, the quality of the light changes. All the green in the leaves and the canopy have essentially absorbed the majority of that good red light. What's left over is this far red light that ekes on down to the forest floor. That far red light hits a different isomer of a phytochrome and changes the cellular response. And we'll look at this in just a quick second. But that's really cool. Plants that are below the canopy, that are shaded, that are outcompeted for red light, receive far red light. That far red light then initiates an entire sequence of events that leads to growth upwards. In a sense, the plant is striving or growing towards the light. It's interesting stuff. I hope that makes some sense. Now, let's take a look at this. So again, you've got this balancing act between red light and far red light. Now, I've removed some of the details in a sense. During the day, PR is converted to PFR, or far red, and then that far red isomer is degraded by enzymes at night. And so in a sense, and again, this is where things get really tricky, and hopefully you don't need to know this for the exam, but you've got this relative buildup of far red, of phytochrome R and phytochrome PFR. And as those two isomers of this protein build up in relative concentrations, that's going to trigger a response from plants. But I can't imagine that test developers are going to ask you that directly. They might tell you about that. They might give you a long prompt, but you do want to recognize that these two isomers are in dynamic concentration within the cell, and as their concentrations go up and down, the plant is going to respond differently. Now, I said I wanted to look at a couple specifics. Let's do that real quick. Boop, 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 boop. Boop, 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 boop. Hop. Everybody's doing good, doing well out there. It's a lovely Sunday afternoon. I'm just kind of killing time. I think I've talked a lot. I'm going to have a sip of water here before we look at some examples. Here is it's kind of a plant joke. You ready for this? This is I got to give credit to my good friend Peter Knierim, filmmaker. Here it is. Where do cantaloupes go in the summertime? Where do cantaloupes go in the summertime? The answer to be revealed in just a moment. Oh yes. Yes. Look at this beautiful slide. Man, he's still talking about cell signaling. He's been here for an hour. He said he'd talk about it for 15 minutes. He's a liar. Yes, I am. But, and I apologize for lying, it just is. Back to your good old signal transduction pathway from like back in 2016 when we started a screencast. Reception, signal transduction, and response. Here, check it out. Not only is it cell signaling, it's going to be freaking second messenger. So here, this plant is bathed in, dang it, hang on. Freaking, I had this thing so dialed in, and then, I didn't test it, so it's my fault. Get out of here, you. Get out of here, you. So fancy. I'm so fancy. All right. Forget about that last image. Here we are. We have got a plant cell. It is just bathed in the full sun. Life is good. This is a little, you ready for this, guys? This is a freaking mung bean that you planted in AP biology class. This is a Brassica rapa plant that you put in the ground in your little styrofoam cell. Or if you're like my friend Lee Ferguson, you put them in deli cups or old water bottles, and you messed with some fast plants this year, I hope, or some mung beans. You germinated some plants. Now, when those beautiful little seedlings, they worked so hard to get up above ground, and eventually that apical meristem comes up, and you have a little bean shoot, and you can almost see the cap on the apical meristem, and if you imagine enough, you can see the cells themselves. Ultimately, you're going to get outfolding of the seed leaves, and if it's a mung bean or a Brassica rapa, it's a dicot, which means it has two seed leaves, dicotyledons. The cotyledons being the starch which give rise to the seed leaves. Actually, the cotyledons are the seed leaves, and the starch inside is going to fuel their development until they can finally break above ground and emerge. Now, if you took the 2013 practice exam, you probably remember that question about putting the seeds in the dark and putting the seeds in the light and seeing what happened. Do they turn yellow or green? Well, here's the biology behind that question. When plants are starved of light, when those seeds germinate, the seed leaves are yellow. But and that's sort of the natural state, the chlorophyll hasn't been made yet. The chloroplasts haven't been fully formed. And that makes sense, because why would you put all this energy into making leaves when there's no sunlight to absorb? Of course, plants don't think that way. They just have evolved to do that. But as soon as that shoot tissue and those cotyledons more specifically, have been exposed to any amount of full visible spectrum, full red light, it sends off this beautiful cascading mechanism. The phytochrome gets excited by that full visible spectrum. It in turn turns on a second messenger here, cyclic GMP, guanine monophosphate, not CAMP, which sets off a signal transduction pathway, which ultimately activates a transcription factor, which changes the gene expression in that plant. And check it out. The response is de-etholiation, which is a fancy word for turning green. So check it out. Full visible spectrum, that's full red light, activates a second messenger. It also helps activate, I should show you this, a calcium channel. So calcium floods into the cell. Calcium brings specific transcription, easy for me to say, calcium brings specific transcription factors to the nucleus. These two proteins work in concert to change gene expression, and the cotyledons go from yellow to green. It's beautiful. Now, that plant is now growing, and it's got green leaves. And now we're going to shade it up. We're going to essentially block the light from it. Let's see what happens next. If we were to block the full visible spectrum, sorry guys, so kind of monkeying around with this, I apologize. If we block the full visible spectrum, we're not going to do it. The plant's going to have a different isomer of phytochrome that's going to pick up that particular wavelength. And that's kind of cool. I mean, that's something I hope that I've made clear, but it's a slippery idea. Plants have two isomers of phytochrome. One phytochrome picks up the full red spectrum, that is that stop sign red. The other phytochrome picks up that far red or that dusky red. And that's the phytochrome we're dealing with right now. When that phytochrome is exposed to far red light, because all the other good light was taken, that's going to set off a different signal transduction pathway. It leads to a different response. And here the response is not the genes which control greening don't get turned on, but the genes which control the production of auxin get turned on. And those plants begin to grow taller. You can see that here with apical growth. So dig that. You've got genes which are turned on, which lead to all those beautiful changes that happen in cells when conditions are necessary for growing up. You're going to get proton pumps being turned on. You're going to get increased growth factors going, and those plants are going to grow up, which makes sense, right? Plants in the shade tend to I apologize for that. Plants grow up. When they're bathed in the full red light, there's a reduction in auxin concentration and an increase in cytokinins, and that leads to a branching out. So once those plants hit full visible light, they're going to start to grow up, and out. Down here in the southeast, we see that a lot with our pine trees. Pine trees in the piney woods, they're these straight trunks that have grown up, and then ultimately they have a crown above, which is kind of almost looks like that, kind of a reverse bowl. Whereas if you see a tree in an open field, and we have a few of those on our campus as well, they don't have that, and that could be, heck, a maple, a birch, a hickory, whatever. I mean, trees which are in the forest, they've got these long, skinny trunks, and then they tall, and then they've got a crown above. Whereas if you've got a maple tree or a birch tree or a tulip poplar tree out in a field somewhere, sweet gum, I'm looking at one out in my backyard right now, that tree has this beautiful kind of full crown because it's bathed in 360 in a full visible spectrum. So it's like, hey, the light's good everywhere. Let's just branch out. Whereas opposed to when trees are crowded, they grow up until they hit that high quality light, and then once they hit the high quality light, they branch out. And that's Dendrology 101, kids. I hope you enjoyed that. I hope someday that shows up on a Jeopardy for you, because it's unlikely to show up on the AP exam. So I better keep on moving. What's next? How we doing out there, YouTube land? We are at 1 hour and 15 minutes, and we're still going strong. Like I said, we'll be here sometime till, I don't know, next Friday. I'm kidding, of course. Where am I? Oh. You're probably wondering. Where do cantaloupes go in the summertime? They go to John Cougar Melon Camp. All right. We covered this. We uncovered, right? Colin and Elijah, if you're still listening. That's the bigs. Cell signaling was the main thing that people wanted. There were also a few questions about gene regulation. So I'm going to do super quick, and a little bit on chemiosmosis, which I'm also going to do relatively quickly. I'm going to direct you back to Mr. Kuhn's talk from 2 weeks ago, which is probably much more succinct than mine. And I'm going to direct you to subscribe to my YouTube channel and watch a couple videos that I've got out on chemiosmosis anyway. So, or watch Bozeman Science, or watch Mr. Kanufsky's talks on photosynthesis. There's a myriad of resources there. We will look at it, but I don't want to waste your entire Sunday afternoon telling you things that you could do on your own. Now, gene regulation 101. Here's the deal. When we think about, we want to separate this out between prokaryotes and eukaryotes, all right? Bottom line, the College Board is likely, if they're going to ask you about gene regulation in prokaryotes, they're going to be talking about the operon model, and we're going to look at that. We're going to look at that in a minute. If they ask you about eukaryotic gene expression, do not make the mistake of talking about operons. Operons are the exclusive domain of prokaryotic organisms, specifically bacteria. And then if one of my moderators can fact check that for me, I don't believe that archaea have operons. It's specifically a bacterial construct. And briefly, operons allow bacteria to regulate which genes are on and off. Eukaryotes, because our genomes are so much more complex, because we are multicellular, because we have different tissues doing just an enormous number of things at any given time, it is crucial that each cell be tightly regulated. Even though each cell, with the exception of the red blood cells, contains your entire genome, only a small sliver of those genes are actually turned on at any given time. And it's also worth noting that the tripwires and the necessary steps for turning on a particular set of genes, it is super complicated because you don't want a liver cell differentiating into a retinal cell. And conversely, you wouldn't want a neural cell to differentiate into an epidermal or epithelial cell. It would be total chaos. So that's one of those kind of beautiful things about biology. It's that, you've got, each cell's got the entire genome, but there's just, again, this small sliver. Just a few of those genes are turned on at any particular time, and it's the result of those few genes being turned on leads to just incredible phenotype. I mean, being multicellular is freaking awesome, man. You get to do all kinds of stuff at once. Being a prokaryote, see if I do my finger right. Oh, that's how they do it. If you're a prokaryote, it's pretty simple. Life is good. You live, you do binary fission, you die. But you do one thing well. If you are eukaryotic like me and everybody else watching this video, man, you can do a lot of stuff. You can do amazing amounts of stuff. So much more than these guys can. But there's a cost, an energetic cost of being a eukaryote. And if you're interested in those ideas about sort of the evolution of prokaryotes versus eukaryotes, I highly recommend checking out Nick Lane, an author of books like Power, Sex, and Suicide, Mitochondria and the Meaning of Life, or his book Oxygen, the molecule that made the world, and his latest book is called The Vital Question. But Nick Lane is this brilliant guy from England who's a deep thinker on evolution of eukaryotes. He can explain it way better than I can. All right. That's the preamble. What day is it? Monday? No, it's still Sunday. All right. Let's take a look. Take a look, babies. Excellent. All right. Oh, yeah. Now, I spent so much freaking time building out this slide in Keynote, so it was just going to be awesome. Here, guys, we've got an operon. All right. What the heck is an operon? An operon is a prokaryotic construct. It is a series of genes which are proximal to each other along the bacteria's chromosome, and each of those genes codes for an RNA transcript, which is then translated into an enzyme, and those enzymes are all related in a metabolic pathway. It's a pretty slick little system that has evolved. When a prokaryote, a bacteria in your gut, let's say this is E. coli, comes into contact with a particular metabolite, substrate, it has the opportunity to turn on enzymes that can use that substrate. And when the substrate is gone, the genes turn off. Freaking awesome. Super tight, super cool. Great example of control of gene expression. So hopefully that question, well, I can't say hopefully. If that question comes up, you should rock that question. It's pretty darn straightforward. Now remember, just because you know about operons, the question's about eukaryotic gene regulation, no operons. Don't talk about it. Talk about something else. But let's take a look. Here, what I want to look at is this particular operon. We call this the LAC-Z operon. It's essentially the operon that bacteria use when lactose is present. They can break lactose down and get energy from it. It's normally off because this bacteria is hanging out in your gut. No lactose. It's an adult gut. It's not getting milk. Haven't had ice cream in a few weeks. No lactose available. Why would you make enzymes to break down lactose? Doesn't make any sense. So, what's cool is that operon is off. Those enzymes are cut off. Now here's the little bit of complexity. Hey Sydney, text me if there's anybody in the chat or if I've just destroyed everybody's morale and they've left. If you wouldn't mind. Last text I got from you was 40 minutes ago. Let me know where we're at because my wife's computer died and too lazy to find the cable here. Yeah. Anyway, hello. Let me know how we're doing. It's like if I'm talking to myself, I'll just stop. But anyway, now it's the end of the year. This operon's, I'm sorry, I digress. What's slick is that this series of genes is influenced or under the control of another gene, which is somewhere away from the operon. And here, this is our what are we calling this guy? This gene right here, it's not part of the operon, but what it does is it's always on, this gene. And it's open to transcription. The transcript is then translated into a regulatory protein. Thanks, Sydney. Awesome. Appreciate that. You can kind of tell I just got in my wheelhouse too, y'all. That gene is always open to transcription. Therefore, that transcript will be translated into this regulatory protein. Here we're showing that regulatory protein. It looks like, if you play with wooden blocks like I did when I was a kid, one of those, rectangles with a hole, half a hole cut out of it. It would be a bridge you'd throw your Hot Wheel underneath it. It's also got a couple of prongs. You'll notice, friends, that protein fits nicely onto this particular gene right here. And this gene, my friends, is called the operator. This is cool stuff. When we think about genes, we often think about genes being transcribed and translated. This particular gene, discrete sequence of DNA, doesn't make anything. It's just sticky to this regulatory protein. Something about the sequence of nucleotides is such that this protein binds to it, has an affinity to it. And it's a stable system. It's always there. That protein is more likely to be bound to this operator than not. Next door to the operator, my friends, is the promoter region. Promoter regions are muy importante. Promoter regions are the place where RNA polymerase binds, because if you want to express a gene, you need RNA polymerase. Now check it out. Pretty straightforward, man. You guys are like, come on, Reardon, we've seen this, we know what we're talking about, move on. But just in case, there's one person that might get something out of this, I'm gonna teach it to you. RNA polymerase, once it binds to the promoter, it wants to move downstream, or it would likely move downstream along this strand of DNA and then transcribe those genes into RNA. But it can't. There's something blocking it. And that thing which is blocking it is the RNA. I'm sorry, it's not the RNA. My apologies, the regulatory protein. And again, this is lactose. This is the lactase operon. the lack operon. Now, I gotta kind of move over just a little bit. I'm gonna start taking some things away. Now, check this out. Boom, goodbye. What happens when, let me move myself out of the way. Again, I appreciate y'all's patience. Come down here. When allolactose is available, that's going to be present when lactose is present inside the cytoplasm of the cell. And this is where kids this is where students can often get confused. The human being, which harbors the E. coli inside their gut, just ate a bunch of ice cream. Lactose is available. It got assimilated through the lining. Well, lactose is available. It didn't all get assimilated through the lining of the gut. It winds up in the large intestine. You've got hundreds of millions of E. coli hanging out there just waiting to scrape up whatever's available to them. Here, lactose is available. So allolactose, which is an alternate form, another allo meaning a different form of lactose, actually binds to our regulatory protein. Can y'all dig that? And when allolactose binds to the regulatory protein, it causes a shape change in that regulatory protein. It causes this regulatory protein to fall off that operator region. It's, in a sense, this regulatory protein has a greater affinity to allolactose than it does to this gene. So it's going to bind to that allolactose and move away. What's cool is that opens up RNA polymerase to go to town on these lactose operon genes. So now RNA polymerase will move downstream and begin transcribing several RNA transcripts, all of these RNA transcripts, get out of here, you inactive protein. All of these RNA transcripts ultimately will be transcribed into 3 enzymes. Each of the transcripts will be translated, into 3 different enzymes, which work in concert to break lactose down so that the bacteria can get some energy from it. Now, again, I realize that's a long explanation, but it's super slick. So just kind of bear with me here. When there's no lactose, shut the genes off. When lactose is present, the repressor protein is moved away. RNA polymerase has access to the genes of interest. Those genes of interest are transcribed, ultimately translated, and then you get proteins in a metabolic pathway that that ultimately break down lactose and yield energy. This is, I don't know if your teachers have talked about this with you or not. Clearly, what we're looking at here on the upper level, you can't see my arrow, I apologize. If we look at the top level layer of this, I'll use this guy as my pointer now. Here we're at the genome region, right? But down here, when we think about proteins working in concert inside the cytosol, whether it's a bacterial construct or a eukaryotic construct, we call this level of organization the metabolome, metabolomics. And that's a really interesting level of study. For those of you guys that are really interested in STEM and interested in health and medicine, you will get to know the metabolome in grad school, med school, dental school. It's a really interesting place to hang out. This is, in a sense, the metabolome is where phenotype truly begins to express itself. Because, and again, this is an offshoot, but I want to spend a second here. If any one of these genes were mutated in this lac operon, you would, and this gene is expressed, remember there are no introns in prokaryotes, any one of these enzymes that will ultimately be expressed might have a mutation in it. Not always, but sometimes, right? So let's imagine this isn't a silent mutation. This is a missense mutation in this particular gene here, the LAC-Z. Well, if that's a missense mutation here, it's likely that this beta-galactosidase doesn't fold properly. Doesn't mean that it's completely non-functional. It just doesn't fold up exactly right, so its activity might be a little slower. That would lead to an entire slowdown of this metabolic pathway, and again, not destroying the pathway, but slowing it down so much that you might actually get byproducts building up from that metabolic pathway, and then as those byproducts build up, that can lead to changes in the overall metabolism of the cell. And for bacteria, it's probably not that interesting, but for a eukaryotic organism with freaking hundreds of thousands, if not millions, of metabolic pathways available with any given cell, if one of those metabolic pathways is broken down, you get byproducts building up, and those byproducts lead to clinical phenotypes. And it's quite a bit easier to diagnose a disease when you see it at the phenotypic level, as opposed to having to dive down at the gene, mining the gene level, looking for the exact mutation that is causing that phenotype. Again, I spent a little longer here than I wanted to, but it's an interesting idea. You probably heard about the concept of pre-diabetic. So if somebody goes to get their blood drawn and they're pre-diabetic, yes, they're looking at a particular blood glucose level that's over 90 milligrams per deciliter, but they're also probably looking for particular biomarkers that may indicate some tissue damage and from increased inflammation due to this pre-diabetic state. And you guys are smart. You know, it's much easier to treat something before it gets out of hand. So if you can get access to that patient, get them on a regimen that's going to reduce their blood sugar, blood glucose levels before they get full-blown diabetes, that saves them money, makes them more productive, puts less burden on the healthcare system. Life is good. So that's a kind of a classic example of biomarkers. Down here in the southeast, we call it the stroke belts. Got a bit of diabetes and obesity, and we have a diabetes and obesity epidemic here in the country, but specifically in the southeast, that leads to ultimately poor health outcomes, marginal health outcomes for those that suffer from those maladies, and then ultimately it can lead to decreased economic productivity. So that's kind of Reardon talking there. I see using health and medicine as a way to get access to people, to getting them healthier before they get sick, so that they can live a healthier, happier life. But that's all on prokaryotes. Let's look at eukaryotes. And again, this is where things get pretty darn confusing. Fortunately, you don't need to know a whole lot about this, I don't think. If somebody wants to disagree with me on the chat, feel free to. Going to the next slide. Remember that I said this is going to be more complicated and more regulated, and indeed it is. So here we're looking at essentially trying to get our head around this idea of transcription factors and gene switches. If you're interested in looking a little bit deeper at gene switches, one of the finest pieces of media you're going to see on that is called Making of the Fittest, Gene Switches. Go to HHMI Biointeractive and search for Stickleback, Making of the Fittest. It talks quite a bit about how particular gene switches are turned off in particular cells, so that you have a reduction in a phenotype. Here with these little fish called sticklebacks, it's a reduction in these pelvic spines, which leads to increased reproductive success when you live in shallow water. Okay, that's the wrong thing. Come on back, baby. Whoa. Hang on, guys. Get a little crazy. Okay, I'm back. You think I would have figured this out after a couple of days? It's been a couple of days that you've been stuck with me, right? That joke never gets old. Now, sorry guys, let me get back to work here. Remember how in operons everything was connected to each other? It was like everything was just sequential and life was good. Even that promoter region was close to the genes of interest. That's not the case in eukaryotic genomes, especially think about humans, for example. 46 chromosomes, 3 billion nucleotides. Doing the rough math, you're about 65 million base pairs per chromosome, and that's a super rough estimate because I'm sure you guys know the first pair of chromosomes is longer than the ones in 20 and 21 and 22. In fact, that's how they get their names. And you also likely know that the majority, vast majority, you call it 75%, call it 90%, of those genes contained within our chromosomes, they're non-coding sequences. They may be regulatory sequences. They may just be remnants of days gone by, their evolutionary baggage. But there's a lot of space between the genes which are expressed. That's the point I'm trying to drive at. Now, choose some ice here. Here's the idea. because those genes are spread apart, because genes require promoters, and because those promoters require what we call transcription factors, you got all these chemicals coming into disparate parts of the chromosome, and what you see are some bending over and increased stabilization of DNA strands one on top of another. Really interesting idea. And clearly, I don't think that you're going to have to write about that in a particular detail on an FRQ, but what I think you'll get, I've seen it in the past, is a relatively long multiple choice question where they're telling you the scenario about how, and I'm going to blow this up so I can see it a little bit better. You might have a promoter region that's going to have an affinity to RNA polymerase, and that, let's say that gene actually is normal, and so it has a high affinity to RNA polymerase. But because there's more complexity, you've also got all these proteins. They're calling them mediator proteins here. Here we call those guys transcription factors. Transcription factor, fancy way of saying a protein that increases the binding affinity of RNA polymerase. So RNA polymerase needs those transcription factors to sit on this particular promoter and then begin to transcribe the genes that are downstream. Well, let's say that the genes all are normal and they code for functional proteins. Let's say that the promoter is normal, no mutations, and it has a high affinity to RNA polymerase. But now imagine this. These little transcription factors, they're proteins. We know that proteins are derived from genes. What if there's a mutation in a gene which codes for a transcription factor, and then that transcription factor either doesn't get translocated to the right spot on the genome, or when it gets there, it doesn't bind effectively? That's going to create some instability for that RNA polymerase, and that gene, which is supposed to be expressed, may not get expressed at all. Maybe so. Also worth noting is that we've got these, what the figure here calls a DNA binding protein. As I mentioned a little bit earlier, what's really cool about eukaryotic gene expression is quite often you've got multiple regions of the genome which are kind of closing down on each other. And so what we're trying to show you here is this linear piece of DNA up top gets twisted and torted by these DNA binding proteins shown in the middle here. I can just use my freaking head as a pointer now. Here, next to my head. And then what's cool is as that DNA gets doubled down and bent over, there are some other binding sites on these genes relatively far away from the promoter region, enhancer regions we call them, that help those transcription factors bind together and collapse down upon RNA polymerase, ultimately allowing that RNA polymerase to do its job. And it is worth noting that RNA polymerase, while incredibly effective at its job, it just does what it's told. It is not a It's not a thing. It's not a regulatory molecule. It's an action molecule. What's cool is you don't want that thing going off, going crazy, expressing genes that don't need to be expressed. And so what you're looking at in this figure, what we're trying to show you is this tight regulation, very intricate set of reactions that has to happen to set RNA up. Because once RNA is set up, boom, it's going to do its job. And what you don't want are cells producing proteins that they don't need. You don't want growth factors in cells that need to be dormant. You don't want to make enzymes for substrates that aren't there. That's a waste of time. I've talked a lot about that. I'm going to move on from there. But again, back to kind of, if I could go back one slide, just for a quick sec. Go to previous. Operons are fairly straightforward. A few genes connected to an operator with a promoter region next to it, and when that regulatory protein is removed from the operator, RNA polymerase has essentially free access to the genes of interest. Compare that to the eukaryotic model that we just looked at, and what you're seeing there is quite a bit more complexity, more parts in discrete in separate locations. They're not all right next to each other, and you've got a series of proteins which work in concert to regulate the activity of RNA polymerase. So it's not just a one and done. You may have 6, 7, 8 proteins, all of which are derived from different genes, working to regulate the transcription of a particular gene sequence. It's really cool, man. It's just genes on top of genes. It's just how our body works. And again, as you can tell by the gray in my beard, I'm middle-aged. I came up doing biology when all this stuff was I wouldn't say being figured out. A lot of this stuff was figured out in the 70s and 80s, but I went to undergrad in the 90s, and so it wasn't really part of my curriculum. It was not what I studied. Genetics was looking at fruit flies, and we did a lot of crosses with German cockroaches, which I found disgusting. But what's been fun about being an adult is as I start to investigate biological systems earlier, we're learning more and more about how these systems operate. We're learning that this phenotypic variability that we see in all kinds of organisms is regulated by these beautiful, intricate interactions between very small molecules. And again, it's let me just be as clear as I possibly can to you folks that are under 21. Sky's the limit in terms of what we can understand about cells and biological systems and how they work. We're just now scratching the surface about how these systems operate, and you will find an incredibly rewarding career in life science in terms of sort of intrinsic motivation, learning cool stuff. It's not a bad one. It's a great field to go into. What I'm trying to say is that we haven't figured jack out yet. We're just starting to scratch the surface, and it's going to be a great century for biological discovery. And I hope that some of you guys watching the screencast will join the endeavor to figure out how biological systems work. All right, moving on. Speaking of working, none of this stuff can work without, you guessed it, energy. Oh yeah. Ow, 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 ow. So it's time, friends. It's time for a little chemiosmosis. We are rocking on at an hour and 42 minutes, and we will go for another 18 minutes at least, and then I will go use some ATP to move bicycle pedals around. But let's take a look, babies. Chemiosmosis, it came up a lot. Let's take a quick look. All right. Booyah. If any of my students are still watching, they know that I don't talk about anything without talking about the context first. So let's do just a little quick bit of context. Come back here. So I'm sure you've all seen these images in your textbook or in class where they show you molecule A is the substrate, and then you've got enzyme 1 leads to intermediate B, which leads to enzyme 2, intermediate C, and enzyme 3, which leads to product D. Well, I took that model and I tried to put it in the context of cell respiration because I think it's a decent little model. Here, imagine this. We've got glucose as our starting molecule. There's a series of reactions called glycolysis, and glycolysis breaks glucose down into pyruvate. So B right here would be pyruvate, a 3-carbon compound that in the presence of oxygen will be further oxidized into acetyl-CoA. That's a 2-carbon compound. That acetyl-CoA will enter the Krebs cycle, or what we call the citric acid cycle, because citrate is an intermediate of that pathway. And the Krebs cycle is going to spin off electron carriers we know to be NADH and FADH2. And then those electron shuttles donate their electrons to the electron transport chain along the inner membrane of the mitochondria. And the end result of that beautiful process is called ATP synthesis. Now, one brave young person asked, what happens when there's no oxygen? When there's no oxygen, pyruvate is not shunted towards the mitochondria, but remains in the cytosol, where it is broken down further into ATP by substrate level fertilization. Listen to me. Substrate level phosphorylation. Substrate level phosphorylation. The deal is, though, that when that pyruvate is broken down further just to yield some little bit inkling more of ATP, that oxidation is going to yield some carbohydrate, and it's also going to yield a byproduct called lactic acid or lactate. Lactate, now remember, I don't want to get too deep into redox, but ultimately pyruvate Three-carbon compound that's reduced, it's got hydrogen, it's got to be broken down to yield energy. There's no free lunch on this planet. I hope you know that. Energy is neither created nor destroyed, merely transformed, and no energy transformation is 100% effective. So if we're going to break pyruvate down into something else and yield ATP, if we're going to make an energy conversion, some of that energy is going to be lost. And worth noting that hydrogen's got to go somewhere. That hydrogen goes on to lactate, forming lactic acid, or you can see, I don't exactly. It forms lactic acid. I'm going to shut up right there. Forms lactic acid. That lactic acid builds up, and we feel that as an achiness in our muscles and even a burning in our muscles if we're really metabolizing energy a lot faster than our mitochondria can keep up with. But that's what happens. Things begin to ferment. Fermentation leads to byproducts. It doesn't yield a whole lot of ATP, but it does grease the wheels enough to keep the process going until eventually you just cramp up and stop running. Now, it's worth noting we do lactic acid fermentation, not ethanolic fermentation. If we did, the Olympics would be a whole lot funnier, and we wouldn't be here because we would all just passed out drunk, running away from our predators, and that would have been it for the hominids. Anywho, let's look at what happens as we dive into oxygenic aerobic respiration, oxidative phosphorylation of ATP. When oxygen is present, like I said earlier, that pyruvate, that three-carbon molecule, is broken down into a two-carbon molecule. That two-carbon molecule is spun around, completely oxidized into carbohydrate, and it yields NAD and FADH2. And I'm not going to spend a ton of time here. What I want to do is look at a next slide that I've got prepared for you guys. It's kind of a classic that most every kid has seen in an AP Bio class. Let me put my face up here. Now, let me just, I'm not trying to show off, but I've got a couple of YouTube videos, specifically on the overview of cell respiration, as well as a deep dive into chemiosmotic production of ATP, both in cell respiration and in the chloroplast. And so if you're looking at this stream, you can see my channel, RyanReardon71. Feel free to watch those, or again, anybody else's. There's several of us out here that have done a lot of deep thinking about this, and we're trying to communicate those ideas to teenagers. So here's my take on this. That was I don't know, like a decade ago. First couple years teaching AP Bio. I'm like, 12th, 13th year now doing this. I watch kids struggle with this, man. I just watch them just pull their hair out trying to figure this stuff out. And I started looking at these figures, and here, this is a classic, and this here we're on the inner membrane of the mitochondria. It looks like a mattress. That's your phospholipid bilayer. This stuff in this kind of beautiful maze down here, that's the matrix of the mitochondria. And then above that, in orange, that's the inner membrane space. What we can't see in this figure is the exterior membrane of the mitochondria. But this orange space is a super tight region. It's the interior of the cristae of the mitochondria. And everybody knows this. You've got these awesome electron transport chains that are embedded within the mitochondrial inner membrane. And there are thousands, perhaps tens of thousands, perhaps hundreds of thousands of ETCs, or electron transport chains, along each mitochondrial inner membrane. That's what these cells do, baby. That's what these organelles do. They freaking move electrons, they move protons, and they make ATP. That is their role in our cells. And it's complicated. But what I realized is that if I broke chemiosmotic production of ATP down into three familiar concepts, kids might start to get it. So I want to share those with you right now. Let's look at step one. Here we're looking at the transfer of, just down here, over here, put my head down there. Let's see that, the base one. NADH, which came from either glycolysis or the Krebs cycle, is oxidized by protein complex 1. By oxidation, we mean it's broken down. It's going to get broken down into its constituent parts. NAD minus, and it's going to lose a hydronium ion and its associated electron. Now, hydrogen atoms don't hold their electrons very closely, so that electron is liberated from the hydronium ion. So you've got an H plus hanging around, and you've got an electron which can then be moved down the electron transport chain. Bottom line, you guys, at step one, redox happens. things are broken down. Electrons are released, and those electrons are cradled down the electron transport chain to an ultimate electron acceptor, which you guys know as water. I'm sorry, ultimate electron acceptor you know as oxygen, the end result being water. We breathe it out every time that we do respiration, open our mouths. That's why my mouth is dry right now. I think I'll have some water. All right. If electrons are flowing, you're generating current, you've got energy. And everybody watching this video right now knows that. If you've got an electrical current, you can do work with that electrical current. The work which is done by this particular electrical current along the inner membrane of the mitochondrion is the active transport of hydrogen ions from the matrix of the mitochondria into the intermembrane space. So part 2 here, these red arrows are showing you the translocation or active transport of hydrogen ions from a relatively large space down here in Mays to an incredibly tight space here in the intermembrane space of the mitochondrion. And dig this, as long as electrons are flowing, which means oxygen is available, and electron shuttles are available, which came from the breakdown of food. So dig this, as long as there's glucose on one end, and as long as there's oxygen on the other, you're going to get the flow of electrons. And as long as electrons are flowing, baby, those hydrogen ions are going to be pumped inside this intermembrane space. Now, you know this. So imagine you're sitting in class right now. 24 of you, 25 of you, something like that. And then there's a freaking tornado drill. And your teacher says, okay, we got to go to the smallest space possible. And he or she says, I need all you guys to go into the prep room. And you go in the prep room, which is about, probably 20% as large as your classroom. You got 25 human beings in there. You probably want to get away from each other. Well, guess what? When you pack a bunch of hydronium ions in here, they want to get the heck away from each other. There's only one way out, babies, and that is through ATP synthase. This is cool. So we've got redox coupled with active transport, and now check it out. We've got the facilitated diffusion of hydronium ions out of the intermembrane space back into the matrix. So these hydrogen ions are going to flow right along their concentration gradient back into the matrix. What's cool about this is that as those hydronium ions flow through ATP synthase, catalytic sites on ATP synthase open up, and what's happening is it's going to grab an adenosine diphosphate, an inorganic phosphate, and cobble those things together to make ATP, which we see as the starburst down here. ATP is always starburst. It's ATP. So check it out. That's a lot of talk, but what's hip is that flow of hydrogen ions through ATP synthase. That is a spontaneous reaction. It's going to happen, man. Things flow along their concentration gradients. When you've got a spontaneous reaction, you can couple that to a non-spontaneous reaction, taking ATP and inorganic phosphate, two simple things, and coupling them together into something complex. That is non-spontaneous. You've got to have energy for that to happen. That energy comes from the flow of hydronium ions. Why do they flow? They flow because they were actively transported into this intermembrane space, and that set up an unstable condition, a positive charge on one side of the membrane and a negative charge on the other. A decreased pH on the orange side, a relatively higher pH on the maize or yellow side, that is instability. How did we get that instability? We got the instability because the electrons were flowing, and as those electrons flow, certain proteins inside these protein complexes have the ability to translocate hydronium ions from one place to another. It's freaking rad. Of course, the question becomes, where did the hydrogen ions come from? They came from the breakdown, the oxidation of NADH and FADH2. They get pumped across. When they flow back, they're available to this highly electronegative oxygen molecule, which is hanging on to these electrons. And guess what happens? H plus an O with two negatives. Booyah, you got water. We breathe it out. Life is good. And this is profound, man. Eukaryotic cells, virtually all eukaryotic cells have mitochondria. Mitochondria takes a very reactive, dangerous molecule, oxygen, and converts it into a metabolic necessity, water. And while that happens, we get energy out of it. It's beautiful. In fact, I'm going on record on YouTube today, April 20-something, 29th, right? 2017. I'm here to say that chemiosmosis is the most beautiful thing in the world. I love it. All right, I said it. There it is. Now let's talk about what happens inside of plants. And for that, I want to look at some different models. Beep, beep. Goodbye, PowerPoint. Hello, models. Oh, yeah. Is this coming up? This is the one I need. Beep, 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 beep. All right. Man, I'm curious as what y'all are seeing. Let's see if I can blow this up. All right. Howdy. All right, this model's, my students give me a hard time about this stuff because I love this stuff. They're like, whatever, man, that made no sense to me. Here's the deal. You got to use your imagination just a little bit. Let me get my mug out of the way here. All right. I bet your teacher talked to you about photosystems. All right, we're on the thylakoid membrane, by the way. We're down on the thylakoid. We're down on the thylakoid. No. And you are looking at, so I'm trying to get right here. Let's see what y'all are looking at. Tell you what, I'm about to make y'all's day. I'm going away. Boop. Okay. Take a look at the lower left-hand side of this image. What we've got here is the reaction center of photosystem 2. And all you've got next above it is a primary electron acceptor. Now look over to the right. You've got reaction center of photosystem 1 with its primary electron acceptor. What I'm trying to show you here in red is that we got electron flow. So here's what happens, man. Let's see what that looks like. Yeah. When water is brought up to the leaves, which are packed with chloroplasts, which are packed with chlorophyll on the thylakoid membrane, water is available to a photon of light. This photon is 680 nanometers long. That particular wavelength of light, which is red, splits water into its constituent parts. Here simply I'm showing you that oxygen is given off by a byproduct, but that's an oversimplification. In fact, water is broken down into hydronium ions, free atomic oxygen, and an associated electron from those hydronium ions. Those electrons are boosted. They get bounced up. They're held in suspension by a primary electron acceptor where they are able to do work because now they're not in their most stable configuration. They want to fall back down and become more stable. And if your teacher ever took spinach leaves and chopped them up or blended them up with acetone and then shined a light on them, you saw that red pigment. This is red light. The reaction center of photosystem 2, in fact, all that chlorophyll, it selectively absorbs red light, and then those electrons in the chlorophyll actually get boosted. They get excited and they fall back down. When they fall back down willy-nilly or out of control, they fluoresce red. But when they get captured by an electron acceptor, they don't fluoresce red. Because if it's held, it's not going to fall. If it doesn't fall, it's not going to fluoresce. If it's held and then shunted down an electron transport chain to translocate hydrogen ions around, that energy has been consumed. Again, I said it earlier, there's no free lunch. You don't get 2 for 1 with biological systems. If the electrons are used to drive chemiosmosis, they can't be used to drive fluorescence, which is why plants look green instead of red. It's because those electrons are not just falling down out of control, they are moving down a tightly regulated series of proteins tumbling down, and as those electrons tumble down, they generate a hydrogen gradient, much like we saw with in chemiosmosis. And that proton gradient inside the thylakoids is used to make ATP. Now, what's cool about photosynthesis, and I realize this is not my finest hour right now, those electrons are boosted from photosystem 2, they tumble through an electron transport system, and then they are boosted again to a secondary primary electron acceptor. Now, some of those electrons are, let's see if I can move this over. Some of those electrons are shunted towards another short electron transport chain, and then that releases energy. That energy is coupled to an enzyme called NADP reductase. And NADP reductase reduces NADP, the cousin of NADH, which you met just a minute, NAD and NADH, which you met a minute ago, NADP, it reduces that to NADPH. Now, I know this is a subtle idea, but NADP and a free hydronium ion, those things are more simple than a NADPH. What I'm trying to tell you here is that you can't get two simple things to become complex without energy. The energy necessary to reduce this cofactor, NADP, into NADPH comes from the flow of electrons through the short electron transport chain. Now, what's cool is that as NADP gets reduced, sometimes those electrons build up. In a sense, the electrons are moving faster than NADPH can, or NADP reductase can keep up with. Therefore, those electrons are shunted back to the original electron transport chain, creating even more chemiosmosis in the thylakoid. So in a sense, this electron transport chain is used twice by two different sets of electrons. Some electrons are flowing, I would say the majority of the electrons are flowing from the primary electron acceptor, see here on the left, and then some of them are being recycled from this other electron acceptor, which you see here on the right. So what, so what, so what? Here's the so what. The energy, the ATP, I should say, that is generated during the light-dependent reactions is used to drive the Calvin cycle. Further, the hydronium, the NADPH, they're carrying the hydrogen and the electrons necessary to reduce, to reduce carbon dioxide into carbohydrate. To me, and again, I know if I were in class and I had you guys next to me, I'd be a lot more succinct and clear. If you're going to take something like carbon dioxide and water, two very simple compounds, and you're going to make something as complex as glucose, or more specifically, a 3-carbon compound called glyceride 3-phosphate or G3P, you need energy for that. You also need material. You can't turn CO2 into CH2O without freaking H. And you get that you get the energy from the light-dependent reactions in the form of ATP, and you get the hydronium ions from the reduction of NADP and NADPH. And again, I've got a video on this as well where I hopefully explain this a bit more clearly. But the thing that I notice about this is that all this material and all this energy is shunted to the Calvin cycle. So why do plants absorb sunlight to make ATP and reduce NADP? That happens when the sun is shining along the thylakoid membrane. The products of those light-dependent reactions are then shunted to the stroma of the chloroplast, where they react with a series of enzymes that we refer to as the Calvin cycle. And let's take a look at that right now. All right, go away, you. Enough of you. Here we go. So here's a fit image that I built to help my kids figure this stuff out. If this will work, blow this up a little bit. Trying to get this thing to move up. Hang on, guys. There we go. Hello, Calvin cycle. Sorry, guys. This is painful right now. I'm trying to get the Calvin cycle in view, guys. That's what I'm going for here. It's just this is not working for me. Blow that up, just right, Goldilocks style. Yes, no, maybe so. Shit. Sorry. Sort of. All right. Okay. I'm going to keep monkeying with this, but for now, we want to realize that CO2 has been diffusing into the plants through the stomata, and now it's at the stroma. And the plants the chloroplasts are they're ready for it, man. They are ready for it. I have no idea why this is not big enough. Let's see how that works. It's about time for another joke. Oh, getting closer, baby. Getting closer. Yay. Hey, there we go. Thank the maker. All right. So we made all that ATP, and we got that NADPH from the light-dependent reactions. I'm going to be over in about 5 minutes, Sydney. I know. I'm planning on this to be over very soon. Here's something that I want you guys to know. This Calvin cycle, this fixation of carbon dioxide into sugar happens in the stroma, and it happens thanks to such a cool enzyme. So enzymes are proteins. Proteins are purple. This particular protein happens to be ringed in gold because it is the most regal of all. Some say it's the most abundant protein on planet Earth. It is rubisco, ribulose bisphosphate carboxylase oxidase. Big long name, important enzyme, because what this enzyme does is it can actually hold inorganic carbon and set off a cascade of reactions that convert that inorganic material into organic carbon, which we know is carbohydrate. Super cool. So as CO2 diffuses into the chloroplast, rubisco, this enzyme, cobbles carbon dioxide to a 5-carbon sugar called ribulose bisphosphate. That, we now have 5 + 1, that gives us a 6-carbon intermediate, which is reconfigured into another intermediate, and it's basically rearranged because ATP is available from the light-dependent reactions. The intermediate is called glyceraldehyde 3-phosphate, which is further reduced into what's called a triose phosphate because there are hydronium ions and electrons available from NADPH. And what you get in the offshoot is an oxidized version of NADPH called NADP and an inorganic phosphate. We can call that a triose phosphate, a 3-carbon sugar with a phosphate group attached to it. This triose phosphate, if you remember, phosphate groups destabilize molecules. That triose phosphate is shunted out of the Calvin cycle, and ultimately it's going to be cobbled together with a partner to make glucose. But what's slick is the majority, in fact, roughly 84% of that triose phosphate is recycled. Inside the Calvin cycle, you've got 5 of them, okay? 5 triose phosphates. They are converted into 3 ribulose bisphosphates. So check this out. I'm going to come on back here for just a quick second. Bring my ugly mug back in here. Hello. And I appreciate you guys' patience. If I've got 5 3-carbon compounds, and I want to make I've got 5 3-carbon compounds, and I want to make 3 5-carbon compounds. Those 3 5-carbon compounds are more complex. They are more complex. Bioenergetics favors them less. They're less likely to come together. So the bottom line, it's kind of thinking about it here on a Sunday afternoon. You're more likely to have 5 3-carbon compounds in an organized fashion than you are to have 3 5-carbon compounds. All right. So the bottom line, to convert 5 3-carbon things into 3 5-carbon things, that's a non-spontaneous reaction. You need energy to make that happen. That energy comes from ATP. That ATP actually comes from the cyclic flow of electrons in photosystem 1. And again, that's why I spent a little bit of time there, because I think that's kind of interesting. Now, so I should make myself small. That's much better. So check it out. And this is it. Last couple minutes here, you guys. CO2, which is simple, gets connected to ribulose bisphosphate, which is more complex than CO2, but together they are less stable than these intermediates. Actually, I take that back. Rubisco can turn that 1 carbon and 5 carbon compound into an unstable 6 carbon compound, which breaks down. But in order to break that unstable intermediate down into 3 carbon sugars, you've got to have ATP. You've got energy to convert one molecule into another molecule. You further reduce those molecules using NADPH, which we got from the light-dependent reactions. That makes a triose phosphate. That's the bedrock of all the sugar that gets made by plants, transported as sucrose and stored as, let's say, amylose and cellulose. The vast majority of that sugar is retained in the Calvin cycle. It's a 3 carbon compound, and it is converted into, there are 5 of them, and they're converted into 3 5 carbon compounds. So basically we're taking something simple, making something more complex. We need ATP to do that. It comes from the cyclic flow of electrons. We're back to ribulose bisphosphate, and the plants are ready for the next molecule of carbon dioxide. Bottom line, look on the right, here we're just above my head. This ATP comes from the non-cyclic flow of electrons. This NADPH comes from the work of NADP reductase. This ATP over here on the left comes from the cyclic flow of electrons. Together, all 3 of those reactions that happen on the thylakoid membrane, those products work in concert inside the stroma to convert carbon dioxide into sugar, which is used by the plant to fuel their own mitochondria, or if there's excess, it's going to be transported and eventually stored. And that, my friends, is the story of chemiosmosis. And to that end, I'm finished. If we look back on what we talked about today, we talked a little bit about cell signaling in terms of transduction pathways, the work of the kinase molecules and their partners in crime, the phosphatases. So proteins which are normally stable, unreactive, get activated by a phosphate coming from because the kinase adds that phosphate to them. That unstable molecule reacts with a partner downstream and then is made stable again. So you've got this cool on-off switch thing happening with a signal transduction pathway. We looked at the results of those signal transduction pathways in terms of long-term response to hormones or growth factors. And we also looked at relatively quick changes in terms of epinephrine being dumped onto the pacemaker of the heart. We also talked a little bit about how plants can respond to the environment using phytochromes. And those phytochromes are sensing different wavelengths of light. And those phytochromes send off signal transduction pathways, which then cause long-term changes and even some short-term changes in plants. We shifted gears, talked a little bit about operons, which are fairly straightforward. And we talked about the intricacy of gene regulation in eukaryotes, thinking a little bit about how there are many redundant pathways, many tiny steps that lead to RNA polymerase actually doing its job, because once RNA polymerase binds to the promoter, it's off to the races. And then we backed out from that, and we looked a little bit about the drivers of all the beautiful biodiversity you see on the planet, chemiosmotic production of ATP from mitochondria, as well as just a brief overview of chemiosmotic production in chloroplasts, and ultimately that energy being converted to sugar. I realize that's a lot to take in. Again, I've got some films or short videos. I know Bozeman Science has got great videos. I bet Mr. Konufke and Mr. Kuhn also have great videos. There's stuff out there. Watch it, but don't knock yourself out so much trying to cover it. We've got another talk next weekend. We've got lab skills with my good dear friend, Ms. Lee Ferguson. And then another good friend of mine, Helen Snodgrass, is going to be talking about last-minute tips and tricks. Stay tuned. I appreciate y'all's time and attention. And with that, Mr. Reardon is signing off from Birmingham just before the storms arrive. Y'all take it easy.